Hey, Dr. Murray, how are you?
I'm well. How are you, Rich?
Good, good, good. So a lot of people coming over from Marshall's presentation. He did run over a couple minutes. Um, so he's the director for the Neuromechanics Laboratory, University of Nevada in Reno. And I'll just turn it over to you, Dr. Murray. Take it away, my friend.
Awesome, awesome. Well, I'll give people here a sec to get in. I'm
Good,
going to share my screen.
good idea.
Make sure y'all can see my lovely PowerPoint. More so, last night, rush job. It's all good. No. Awesome. That should be up for you guys. I'm seeing it on my end. If you can't see it, please let me know.
It's all.
Okay. All right, folks. Well, pleasure to be here. I really, really appreciate y'all coming today. I appreciate everyone's hard work in kind of putting this event together, especially Tekscan for inviting me to be here, and Marshall. We go back quite a few years, been friends for quite a while now, and so it's a really cool opportunity. I actually met Marshall back when I was a PhD student in UTEP, and it's carried over ever since then, and now here I am talking to you guys, and I'm pretty darn excited to be here. So a little bit about me before we get going. You can see our beautiful campus right over here, and what we've got is, if you've ever been up to Reno, you should, because it's way better than Vegas. Everyone knows where Vegas is, and everyone wants to go to Vegas, but you should come to Reno instead, because it is 10 times better, because it's actually pretty. It's in the mountains. We have Tahoe nearby, so we really have an awesome opportunity here. And again, as Rich said, for those of you who are just joining us, my name is Dr. Nick Murray. You can call me whatever you'd like, but I am an assistant professor at the University of Nevada, Reno, in the kinesiology group here, part of the School of Community Health Sciences. And I have a pretty cool and awesome, unique role that I fulfill here outside of teaching. I do provide concussion management services for folks in our surrounding area. So our lab services all the high schools, all the sports teams, professional, semi-pro, all those fun stuff. And I'm kind of that person that allows us to do independent evaluation for people when they have a head injury, working alongside their physicians and physician groups to make sure that they are okay to go back to play and return to play. So we have kind of a fun opportunity here. I'm going to use this session to talk a bit about how we can relate center of force to center of pressure. And then I'm going to be talking more in depth about the outcomes relating to what does this look like with sport-related concussion. Since that's my focus, my area, I want to spend some time giving that some direction and some attention. All right, so just to give a couple disclosures right now, I am funded by National Institutes of Health, and we do have a couple grants set up. There's the grant number if you'd like to take a look at it. It's through the Neuroscience Cobre. I'm part of the neuroscience group here as well. And if you'd like to take a look, we got our full publication list that's available. I'd like to put my Google Scholar out there. I'm also on ResearchGate if you'd like to check it out. There's a lot of different opportunities to get in contact with me. I'm not shy. So I also put my email at the end. I would love to chat more with you, hear your thoughts, and anything. I mean, realistically, I'm an open book, and I would love to hear more and discuss things with you guys. And we have collaborations all over the country. Right now, we're doing different projects. One of them is actually we are doing muscular dystrophy research on top of our concussion research, and we do this all over the world now, which is really, really cool. But we do that with remote technology. So what I'm going to talk specifically about today is I'm going to talk about this concept in biomechanics called center of pressure. Walk you through some of the nuances, how that differs from center of gravity, and why it's important for us from a neurological consideration, and why we use it very frequently to assess any type of neurological disorders. And then I'm going to move into talking about how do we collect it from the traditional standpoint, alongside using the Tekscan stride way to do an analysis using the center of force and transferring that to center of pressure. Then I'm going to review some basic examples just to give you a understanding of, okay, what does this look like in a real world setting? What does this look like? And again, I'm going to focus on our sport-related concussions so you have a better idea of how you can see and use this information. All right. So let's first talk about some of the theory behind postural control. And one of these things that we have to consider is the inverted pendulum. So as most of you are aware, as I'm seeing, we have physiotherapists, we have MDs, we have all sorts of folks on this call, which is fantastic. And I guarantee you most of you know this already and are probably more knowledgeable than I am in this. However, we need to kind of discuss this from the basic nuances so that we understand what we're measuring. The inverted pendulum tells us that 2/3 of our total mass is really located above our belly button. So I have this really awesome stick figure that I drew. And this dashed line gives you a representation of, we've got this big section here, head, arms, and trunk, that contain the majority of our mass. This is actually a good thing for us as a human system because it allows us to have increased mobility. We are a mobile creature. We want to be able to move quickly. We want to be able to move out of the way of a car as it comes. We play sports. We have multiple degrees of freedom so that we can then adjust to course directions really suddenly. And that's why, for the most part, this is an advantage for us, but it also does create some issues when we're trying to control for movement, especially in pathological populations, neurological disorders, Parkinson's disease, stroke, cerebral palsy, concussion, these different areas. It does create some difficulties when you can't control that movement. And so you end up with some angular momentum that the body creates during standing, and more specifically when you're walking. You have this inverted pendulum that shifts you backwards and forwards, and we can most of the time use that momentum to our advantage. But if you have deficits within the neurological system, whether it's from a basic muscular imbalance, basic muscular issue, there is some sort of a disadvantage that can come with that. And so we want to study what this angular momentum kind of tells us about the human system and how well we can adjust for it. And at any point, I put my citations down in the corner. You can email me for a full citation list if you like. But again, I like to throw out the godfather of biomechanics, in my opinion, David Winter. I had the express privilege in my PhD to meet him, and one of the nicest men that I've ever met. He's no longer with us, but goodness gracious, this man pioneered the field of biomechanics. I don't think I would be in biomechanics if it wasn't for folks like him. And so all those who come before me, thank you. You guys are awesome. All right, so let's go ahead and talk about this concept of center of pressure. So now that we understand the inverted pendulum and how this moves relative to one another, we have this concept. This total summation of the force, and we're really applying Newton's laws, and specifically we're looking at vertical ground reaction forces and how these play a role. So why is this important? Well, okay, we also have this concept called center of mass, and I'm going to use these two concepts here to help us explain what we've got. But center of mass in general is the central point at which the body kind of oscillates relative to gravity. And I've got a nice little picture on here, but you can imagine just a plumb line through the center of your body, and if you're quietly standing right there, your body, the center of gravity acts through that central point, and we can measure a lot of outcomes based on the center of mass and center of pressure relationship. So what is center of pressure in relationship to this? So in more graphical words, we've got our little guy here again, and I have some feet print over here, but we're going to start over on the left side here first. So here's our little human guy. I've drawn a plumb line for you that's basically acting to the very center of the human system that is in coordination with gravity, okay? Now, that's what we call the center of gravity, center of mass, interchangeable constructs at times. And so on the inverse area, what goes down, and we apply a force directly to the ground, there's an opposite and equal reaction from the ground pushing back on us. So we have this purple vector here that I've drawn, which is the vertical ground reaction force. This, if you can imagine, and I always kind of give it to my students, if you're sitting in a chair, this is the main reason why the chair doesn't fall through the ground, or you don't fall through the ground, because the ground exerts an equal and opposite force to you as you're standing, sitting, however it is. Well, we can use the vertical ground reaction force to get us an idea of pathological issues. On the right-hand side here, we've got some footprints. So if you're looking directly down through your body and you're looking at your feet right down, you got your left and your right here, I'm going to say that this little red dot here is your center of gravity or your center of mass. Now, as you move, it changes its position relative to the center of gravity or how far you're forward. So if you displace forward, that center of gravity is projected forward. Here's a nice little example of it again. Yes, these are all my own slides. I made them. They're super fancy. No, they're not. They are just what they are. But here's the displacement here from inside the base of support, moving out and displacing. So this is somebody who could potentially like reaching forward and moving forward. You've got that movement there out to this portion. Now, what the center of pressure does is it extends and exerts farther than the center of gravity. So it goes outside the center of gravity and corrals and pushes the center of gravity back into the base of support. So it works in this way, where if you move forward and you're getting to a point of instability that you may fall, all right, the center of pressure pushes the center of mass, center of gravity back towards the base of support, so you limit the possible interaction of falling. So the basic sense of mind, if you move and you displace, the center of pressure moves you back into that safe space so you don't fall and prevents for that. Well, we can measure these constructs relative to one another, and we typically use this center of pressure as a control mechanism. It kind of describes how well and how accurately we can control our upright stance, our movement, or however it is. So this is from a 2007 study where we look at this relationship between center of pressure and center of mass, and it's one of the main reasons why we can use the center of pressure as a calculation to describe the human movement. I've got a nice little figure over here, and you have this black line here is going to be your center of pressure, and this gray line here is your center of mass. You have a couple different positions. So just upright standing, and then you're leaning forward, and you're leaning further, leaning farther. And what you're doing is you're moving that center of mass out in front of you, and you can see how the center of pressure exerts farther and moves alongside the center of mass. So if we were to run correlations on this data, it's nearly a perfect relationship, anywhere from 0.9 to 0.98, depending on the activity. And the more dynamic the activity, the more near perfect relationship that you actually get from a correlational component. And so you can kind of see how it pushes it back in and controls for that relationship and allows you to have the ability to be flexible and move yourself relative to how the center of mass moves. I like to put this, and this is just because I spent some time on a farm growing up, and this is how I learned it. One of the easiest ways that I teach my students, if we're getting stuck in the muddy details, is you can think about sheep and sheepdog. And I don't know if you've ever seen some or had the opportunity to see one. All right, what does a sheep and sheepdog do? Well, if a sheep goes left and there's a cliff there, or there's something that they want to do, the sheepdog runs outside the sheep and barks, and pushes them back towards the opposite direction they want to go. So the sheep in this context are essentially the center of mass, and the sheepdog is the center of pressure. And so the sheepdog corrals the center of mass back into the base of support. All right, so traditionally, how do we measure this? Well, I'm sure most of you are familiar with force plate technology. Some of us have this in our lab, some of us don't. Some use different types of platforms. There's many different ways to measure it, but I'm going to start the most basic way that we typically do this. And this also has some advantageous aspects, but limitations to it. So first and foremost, how do we measure it? We typically measure it with force platforms. Okay, these force plates that are embedded in the ground, and as an example of, these are not movable. They're really highly sensitive to noise. I don't know if anyone's had the opportunity to visit any of the University of Omaha, Nebraska centers with Dr. Sturgio and his group out there. They have specific rooms just dedicated to their force plates, completely isolated from everybody else. All right, that's useful, but it's also difficult. And it's difficult to use that and to build something like that. And so we have to look for different ways to accommodate can we embed these plates in the ground into surfaces that are not going to move. A lot of the time needs to be on the ground floor. There is some technology that allows us to filter out building sway now, but it becomes difficult. In our lab here, we had to do some innovations when I first started about 3 years ago. And we had to actually dig down all the way 8 feet into the ground to build it back up to create a level perfect station for this. And it's difficult. It's not easy to do. Cost decent amount of money as well. They are excellent, though. I mean, they really give us a lot of great information. And so these force plates can be quarantined into looking at what happens in an anterior-posterior direction, medial lateral, and a vertical direction with different ways of measuring that. And so this is the most common tech that we have, I would say. And there's advantages and disadvantages. So from the most basic perspective, I've got a couple sample equations here. And I'm not a mathematician by no scale of means. I can do the basics. But in general, there are multiple ways to calculate center of pressure, but they all kind of come back to this major factor. This is actually a sample center of pressure equation from actually Robertson's book that I used for my grad course that I learned how to do my center of pressure evaluations for using an AMTI force platform. There's multiple different manufacturers, and they sometimes have different equations. I know Bertic has a different equation, so does Kistler. But at the basic level, AMTI kind of has this equation here. So we have center pressure X, center pressure Y, and we have these different scaling factors. But the important thing here is that we have a force in a specific direction. So in this case, it's X. In this case, it's Y. And it's divided by the vertical ground reaction force. And then you also have these free moments, or essentially the torque. And so there's a rotational component that allows us to measure how far it moves, excurts based on a rotational component. You can see this, and there's different scaling factors that are involved here. So again, we have to, each plate is different, has different outcomes, but we have to measure this with these particular equations, and you can kind of break them down even easier if you'd like to. But again, this is the basic center of pressure equation. So it gives you an idea that we are, again, dealing with how far we moved relative to a vertical ground reaction force. And so we are using a displacement in a specific direction, or a force vector in a specific direction divided by a vertical ground reaction force. All right. Here is, at the basic outcome, an example of a center of pressure scatter plot. I like to call it a spaghetti plot because it literally looks like if you took a plate of spaghetti and you threw it on the wall, and that's what you get, okay? A little bit of blue. I don't know what the blue stands for, but heck, we can roll with that. This is actually sample data from our lab. This is 30 seconds of quiet, feet together, upright standing, eyes closed on a force platform. And these lines are just the trace of the raw movement of the center of pressure in X and Y direction plotted over one another. A lot of times in the research, we dichotomize out and we take a look at COPX versus COPY. We want to look at them because they have specific strategies. COPX indicates movement in one direction. COPY indicates movement in another direction. But in this very basic outcome you can see here, red is a trace of a sport-related concussion. That's what SRC stands for, the sport-related concussion. And blue is a healthy normal control. And in general, it doesn't look hugely different. There is some excursion aspects on the corners here that you can see in general in this X direction, that they sway more. And so sport-related concussion using center of pressure, in general, they move more, they sway more, and we can measure that with center of pressure, and we can apply other mathematical algorithms to it. We can run root mean square, we can run some of the basic just total excursion, confidence ellipse, velocity. You can do acceleration, not as useful, jerk, those type of things. You can use the different orders to get an understanding of the human movement. And one thing on here that I'll kind of mention a little bit is it's actually kind of funny when you use first order derivations or just simple how much they swayed, so excursion or path length. You don't see that much with sport-related concussion. It's actually a lot. You see a lot of changes in the velocity metric. They become way faster with their course corrections and their postural corrections than they need to. But this is just a basic example of what this ends up looking like from a raw data perspective. Ooh, my mouse is running slow. There we go. Another way that we can use this, so I'm getting to the point where we're going to start using gait, and we're still on force plates. So you can use this for posture. You can use this for gait. We've been using this for years and years and years, and plenty of excellent research has come far before me, have done multiple aspects. So one of my collaborators here, actually at the University of Delaware, Tom Buckley, we've been working on taking a look at gait initiation and gait cessation, or how you start and how you stop walking. We look at that in sport-related concussion, and we notice some aspects. So this is actually a trace from our recent article. And if we got this black trace here, this is going to be your sport-related concussion at actually what we call this clearance. So this is return to play when the person is going back to play. And then this is their baseline score in general, or their baseline center of pressure. So we have medial center of pressure displacement on this x-axis, and then anterior posterior center of pressure displacement in x and then our x and y. So in these phases, I'm not going to get into the nuances of it, but this is really to show you that we can use it during gait. We can measure these during gait. But again, this is on a force platform. So if you're going to measure this, they have to start and end on a force platform. Well, do the math a little bit. If each force platform costs anywhere from $12,000 to $15,000, you're ending up with typically, an investment of 60 to 80 grand, depending on where you're at, to do this type of research. And that's costly at times. And so very sensitive, very useful, gives us a lot of information. And in this population, specifically sport-related concussion, they typically exert less, they have less displacement. And again, this is during gait initiation. This is the stepping pattern that we see, the loading and unloading in a specific direction. They have a different pattern, a more conservative gait pattern. They don't excurt as much, and that's usually because there's a lockdown effect going on in the body. But again, this is an example of how we can use this in just gait rather than just posture quietly standing on a force platform. So there's multiple aspects of how we can do this. Now, in terms of movement strategies, I think it's important to note that from a postural perspective, or how we control our, remember that hat, that two-thirds of our mass located above the belly button area, that angular momentum, there's quite a few different ways we can use it. And this is why it's useful for not only musculoskeletal disorders, but also for neurological disorders. We have this concept called feedback control and feed-forward control. Feedback control, in general, is from ground up. So we're talking our senses that allow us to feel the ground, get the pressure, and it's a very reflexive loop. And so if you're just basically standing there, you don't put too much effort into quietly standing unless you remove a sensory system, and you kind of sway a little bit and you move. But if you move too far one way or the other, the body creates some sort of a muscular contraction to push you back into those bases of support. So that's the first line of defense that your body has, typically using ankle mobility, ankle extensors to kind of control for that basic movement. From a anticipatory control aspect, this is when things get more complicated. So as the task increases and becomes more difficult, we use higher brain order centers and cortical areas in general to then do top-down control, which involves a lot of like initiating movements, stopping movements, sport movements, trying to discern what object you should grab in the environment relative to a specific outcome, and being able to ambulate, move, anticipate what's going to occur. And during posture and gait in general, it's kind of a trade-off between the two. We want to kind of use a lot of reactive outcomes rather than anticipatory, because anticipatory is more exhausting in terms of the cost for the entire human system. But there's two different ways that we can do it. And again, I'm showing you this because our sport-related concussion example that we're going to get to here, there's a lot of issues with our ability to formulate before, we're not going to talk about those now, but in general, you're walking and you walk with that heel to toe gait pattern, it definitely has some different outcomes, okay? And we try to measure those. So what I have here, now I'm going to switch over from talking and using just the force platform, I'm going to switch over to center force, okay? I'm going to look over at the stride way. And if you haven't used a stride way before from Tekscan, you've seen it, it's basically a series of interlocking mini force plates. I'll put it that way. And they slide together and they have stem cells, billions of stem cells underneath them. And you can lock them together and actually do really cool specific configurations. And they put together, I think our walkway is 12 or 13 feet. So I think it's like 5 different tiles. And so I have some examples of what you would typically see in a normal gait pattern, okay, which you would normally see if this was in like a clinic. So I have some examples, and I have some videos. Hopefully it shows up on your end okay. If not, I'll play it a couple times. Here we go. So what you're looking at here, and I'll play it again, this is somebody walking on the strideway, and you're seeing the formation of the feet. You're seeing the outline of the pattern of the different footfalls. And we're probably really familiar with seeing this. If you've done anything in orthotics, podiatry, those type of fields, you'll see this movement. Here's what it really looks like when you actually analyze the data. So when you look at all the frames together, you get these footfalls. You've got left and right, and our system only allows us to capture a certain amount of footfalls because it is only so long. But you're able to see these individual pressures and analyze this data. And you create stripe boxes using the Strideway software, and it allows us to get spatial temporal characteristics along with force characteristics because it is a pressure-based system. So you're probably really familiar, if you haven't seen this already, you get this human gait table. So in this circumstance, we had 4 footfalls or 4 strikes, and we were able to calculate the cadence of this person, how much time they spent in the gait cycle, the total distance they traveled, the gait velocity. This is all things that are, pretty run-of-the-mill for any type of a pressure mat system. I will give a shameless plug because obviously it's Tekscan's product. It's one of the best on the market that I've had the opportunity to use. I've used a lot of different ones. This one gives you a lot of really useful information. This is only one table of, I think at times, almost overwhelmingly like 30, 40 tables that you can pop up and get all kinds of cool stuff. But it's very useful data. And so we use this frequently with our muscular dystrophy patients, for example. So when we're doing any type of interventions or we're studying the human movement, or for us right now, we're studying the decline in muscular dystrophy, potentially to move a drug to clinical trial, we measure some of those specific outcomes. And so we use our Strideway to give us some good idea of, okay, how is the gait cycle changing for these folks? So that's what you'll normally see with a gait pattern, a very typical normal gait pattern. All right, so this is what it looks like now if you were to do tandem gait. You see how messy that is? This is actually someone doing tandem gait here. You can see the left, right, left, right, left, right. And then they go to the end and they physically turn around and they come back. So tandem gait is a pathway that goes down and back on the same center. Well, this creates complications with the analysis. We can't really generate these nice strike boxes and take a look at each one of the individual footfalls. So we use this thing, this construct, this line that's here called the center of force. And the center of force is very close to and mimics what the center of pressure is attempting to do during the human gait pattern. And it follows a specific line of trajectory during the walking pattern. It's not the perfect one-to-one relationship, but you're able to access this information from the system, especially if you're doing multiple passes, people turning around, any type of assistive devices too. You can remove those other strikes on the system or remove those footfalls, but at times it's really heavy on the post-processing. So if you're in a clinical setting and you need to get data now immediately, sometimes it's a little too cumbersome, and you have to have the people walk on the strideway in one specific direction all the way through. And so there's ways to kind of compensate for this and get around to that. All right, so noisy data. And what we can extract from the center of force is typically it comes out in the send cells, which row and which column are specifically active based on a direction, so medial, lateral, anterior, posterior, alongside vertical. And when you export it out, it looks something like this. So you can export directly from the strideway to give us an idea of time and specific frames, and each row and column and total raw sum of the forces being distributed across the footfalls. So we take this specific data here and we're able to analyze it with traditional metrics for center of pressure. Now again, not the exact one-to-one thing, but we're getting pretty close here with center of pressure versus center of force. They essentially can be mimic and mirroring it. We need to do some more research on exactly the full relationship between the two, but this is a viable measure of being able to extract this on the strideway without having to have a force plate. You have multiple force, like the strideway has multiple tiles set up together, but allows us to measure these as it goes. So what does this look like? When we calculate these out, we have these raw extracted values, and I just plotted this here for us, and so we can see here that we have anterior posterior movement, and now it's going up and it's going back down because this is tandem gait. They're starting at 0 and ending at 0, so 0 being the start of the strideway all the way to the end. So you could calculate this and you could crop it here if you need to do just an analysis on anterior posterior movement. But more importantly, you see this now? This is the medial lateral movement, and you can actually see this medial lateral sway as it begins to oscillate around this centroid portion. This data, but this is the first pass of tandem gait, this is the turn, and this is the second pass. And so it allows us to discretely analyze based on either video verification or whatever way your lab has a good setup to do it, and say, we just want to take a look at what happens in their first pass when they're first walking down versus just the second pass or the turn. And a lot of the times, if you've done work with sport-related concussion, they have an issue with turning in general. Actually, some of my colleagues that have done fantastic work, like Peter Fino, David Howell, these other groups all over the country, you know, concussion, it's a smaller world, but at the same time, it's a pretty unique world, and there's a lot of us that like to collaborate together. And a lot of these folks, again, are significantly better researchers than I am, and they provide us with some templates on how to do these things. And so this turn here is actually the turning synergy turning sequence is one that is quite complicated using executive function and top-down control. And so we like to study a lot what happens during the turns. And the strideway using center of force has allowed us to measure that, because before we have not been able to physically get those turning sequences measured correctly from stride boxes or any of those type of traditional gait measures. All right, so I'm going to make a quick jump over here into sport-related concussion and why we study it, and we're going to look at some of our data from our recent publication on this. So just for those of you who are not aware, sport-related concussion is in general a metabolic disissue, not a direct cell damage. Actually, as a matter of fact, if you were to, sport-related concussion is also synonymous with mild traumatic brain injury. If you measure these outcomes and you do MR scanning or any type of imaging on the brain itself, you're not going to see much. You're going to see the brain actually lighting up like a Christmas tree if you do fMRI scanning. And here I have a Dti scan, and this is actually from one of the pediatric neurology. And so if you look at this here, this is your healthy control water diffusivity that's moving across the brain and across the different hemispheres. You have left, right, and then you have this central portion here, brainstem. You have all this fun activities area. In general, concussions are categorized by this hyperactivity. And so our concussion here, the more red and orange areas means more water diffusivity that's occurring across the brain itself. And so in general, there's more going on in a concussed brain than is for a control, and it's really something that is not helpful, and so this leads to a hyperactivity state. And so post-concussion, you have a reduction in cerebral blood flow to the brain, which reduces the oxygen uptake to the brain. And if you're doing more and you do not have enough energy source from the oxygen to create ATP, you create a what's called metabolic cascade in terms of a imbalance in the ability to have return to homeostasis and to properly regulate the system. This is where the symptoms of a concussion come from. So you have this, everything's going like crazy and you cannot regenerate enough activity. And then at the end of the day, you start to feel really junky. And so I like to explain it. I have my handy dandy water bottle here. Right? Everybody starts with a certain amount of water in their water bottle. And concussions start out with less. And so this is like their energy reserve. And so once your water bottle is depleted, you get more symptoms, typically time to rest. And that's what I tell a lot of my patients, is that you have to take it easy. And so by 2 or 3 o'clock in the afternoon, when you're acute post-concussion, you start to feel pretty junky in general. Postural instability, postural control issues are a cardinal sign of sport-related concussion. One of my advisors, Dr. Kevin Guslowicz at the University of Chapel Hill, he's been studying this. You've heard of the BEST balance air scoring system. He's one of those guys that's just kind of a landmark in concussion research. But we've been studying how postural sway and gait abnormalities occur post-concussion. So real quick, tandem gait. Here it is. This is actually from Jesse Oldham. Jesse is wonderful. If you ever had a chance to meet anyone, Jesse was a doctoral student of Tom Buckley out of Delaware again. But this is the typical setup. It's actually really easy to administer. You actually put a line deer on the ground, and you have a 3 by 38 millimeter line, and you walk heel to toe as fast as you possibly can to complete the task. Once you get to the end, you do a 180-degree on-point turn, pivot turn, and go back to the starting position. So here is what the data specifically looks like. These are the traces from the strideway itself. This is a healthy control here, and this is a concussion. So notably, come on. Let's get back to this one. It's not wanting me to play this. Come on. Right. Nope. And yeah, that's going to be fun. All right. Well, we won't show the video then, unfortunately. I'll show you the still pictures. But what we have here is the healthy again and the concussion here. And here's the traces of it. So what you would have seen in the video here is this healthy person was walking down and completing this tandem gait rather quickly and finishing, and I think they were finishing about anywhere from 8 to 9 seconds. Very quick. Here's our concussion. This particular person, this is their actual baseline. This is their post-concussion. And the traces don't look too terribly dissimilar, but the concussions finish. Okay, this person finished, I think their trial was anywhere, I think it was 17 seconds. So they essentially doubled their time to complete the task because you are doing coordinated heel-to-toe walking. It's quite difficult. And there's a couple things I want to outline and draw your attention to. So I just got some examples here where we measure, in general, this path length of this center of force trajectory and how it moves in the anterior, posterior, and medial lateral direction. Medial lateral being somewhat of the most important in this circumstance. And you can see visibly here, they're not walking on the exact same trace, but they have these little dips here and these movements that we're measuring. We're measuring basically these valleys to peaks during this movement, and they are more exaggerated in the medial lateral direction for our concussion. Not a huge difference, but there's definitely something there. More importantly, if I have you take a look at the turn here, right? Again, this is our healthy person. They turn in point without any issue whatsoever, turn around and keep going. Our concussions, you could visibly see this footprint. I think this is a really cool slide. I did make it, so I'm biased, obviously. But you can physically see this person, instead of doing this nice pivot turn, step, step. And so it allows us to be a little bit more objective with our outcomes, in my opinion, instead of just time, because we measure this in time. And I use that from a clinical perspective to say, like, hey, yes, you were 2 seconds slower than your baseline, or you're 8 seconds past your baseline. Well, that's cool. Okay, that's helpful from just a basic clinical perspective. It doesn't tell us anything about what's going on neurologically. It tells us nothing. Just says you took more time. So center of pressure allows us to indirectly measure how the brain is working and coordinating these movement patterns. And I think that's why it's so useful. And again, I just love this slide. Now, this is obviously one of our more exaggerated cases, but real big difference in the turning sequence here. So on our study that's currently in review in the Journal of Neurotrauma, we have 30 NCAA Division I athletes and 30 closely matched controls. If I had baseline data for everybody, I would have done it, but I will tell you, some of this data comes from our last season during COVID. We couldn't do physical baseline testing. We actually had to do a lot of it remotely in different areas and all sorts of things, so we actually couldn't get all of our folks on the Strideway because it was too much time, too close proximity. Even though we were all in a nice little COVID testing bubble, it still made it really difficult, and we had to keep everybody safe as best as we can. All these concussions are seen in our facility within 48 hours post-concussion. We see a lot of these folks. Again, on average, we're getting right now from all of our collaborators in the county, right now, 5 to 6 a week. So we do see a lot of concussions, and we see pediatric all the way to older adults. But my focus, again, because my love of sports, is on this Division I athletes. All right, so here is the time, just to show you. I'm going to show you time, and I'm going to show you center of pressure. So what this graph shows you, this is the first pass that's extracted, the second pass, and the turn. These are the specific time aspects and how long it takes for them to complete it. So the first pass here is clearly different. It is statistically different too, in terms of time. So it takes a healthy person, a little about 4 seconds to complete it. Concussion about 6 seconds, a little higher than that. Second pass, you see that same pattern. So you can already notice that if you were to add these means together, you're going to get already an increase of about 6 to 7 seconds in general. And then down at the turn, there's actually not a huge difference. And so there's really no difference in the total amount of time it takes for them to take the turn. But the center of pressure starts to tell us more on these specific outcomes. So here's a big old table, and I'm going to highlight a couple things, just a couple things. These are from our article that's in review. FP is first pass, SP is second pass, and then we have turn data. And we calculated from the center of force data, the mean data, and we don't usually do that in terms of gait measures. We do measure the velocity of that forward control, but again, most of the deficits we see are in medial-lateral direction with that hip movement. So here, I want to highlight a couple things. On the first pass, when you are a concussed individual, you excurt more. You have more postural sway in the medial-lateral direction. Same thing, you have more velocity. You have decreased velocity in the forward movement here, and that's typically, again, velocity is a construct of displacement and time, so that's not terribly common. If you take more time to complete something, okay, you're probably going to be a little bit slower and you'll have less velocity. But more importantly, this one got overlaid weird, got messed up. But I'm going to move back from that. I'm going to just highlight just this turn data. So if you can see my pointer here, in the turn itself, the time wasn't significantly different or very varying, but they did excurt, on average, half a centimeter more. That's not terribly clinically relevant, but it tells us that they do have less control, less ability to actually excurt and move in that turn. So when we see that turning sequence with their feet, we know why that's occurring, and I think it's because they're taking more steps in general, and they're needing to take more time to be able to move around that instead of doing on-point turn. Also, if you've ever tried to do an on-point turn when you have a concussion, and if you have any vestibular or ocular symptoms, you get pretty dizzy afterwards immediately. And there's another portion of this data set that I could share with you at another time. That is the relationship between the vestibular ocular motor screening exam and these tandem gait data. And yes, lo and behold, there's some pretty high relationships between if you have vestibular and ocular symptoms and you move more. And again, these are mean data in general. But relevancy-wise, so I use Cohen's d to tell us clinical significance. The higher the value is, the more relevant it is. The velocity is pretty relevant, but again, this turning data here is pretty relevant in terms of its outcomes. So we don't know exactly the full reason why people make this turn differently from a neurologic perspective, but I believe it is something that we're going to continue to study and get a better idea for. All right. Well, I have talked way too long, and I appreciate your time very, very much. And I do want to give a shout out to my team. Obviously, my collaborators, Dr. Islas here, who is our head of our sports medicine physician group here, fantastic. He actually trained me in concussion management, kind of owe a lot of my career to him. My good buddy Ryan Moran at University of Alabama, Tom Buckley, University of Delaware, David Howell up in Colorado, and then all of my wonderful staff that just support me every single step of the way. We have 3 doctoral students, multiple master's students, multiple undergrad students, and we have a good time. If you have questions, I'll go ahead and take them now, but you can also email me, visit our lab site. I'm happy to discuss more.
Sweet. You want to stop sharing your screen?
Yeah.
Great job, Dr. Murray.
Thanks.
A lot of good information. Definitely. Lots of great comments. Definitely a couple questions.
Sure.
See them in the right. We have one. So let's see. There was one up here. How do you consider functional hallux limitus interfering with the center of pressure?
So again, how do I consider functional outcomes?
Functional hallux limitus interfering with the center of pressure.
Good question. If you have less mobility, you're going to have less movement across the foot, and the natural trajectory of that center of force and center of pressure is to actually move along the pathway of the supporting surfaces. So if you have reductions in the degrees of freedom, or you have increases in mobility, either way, like if I'm thinking like navicular drop on top of that, you will have changes in center of pressure, and you will have more excursion that the body has to acclimatize to, which then gives us some issues. Now, not the areas that I study, but you can see if you have less mobility or more mobility, you're going to have increases or decreases in center of pressure, respectively.
Absolutely. What about measuring the center of force? Is it possible to measure center of force with the Tekscan walkway system without the strideway?
Say that last one, Rich. We broke up on the last one.
Oh, sorry. Is it possible to measure center of force with the Tekscan walkway without the strideway? Probably a question for Tekscan.
Yeah, I mean, Marshall, help me out, buddy.
Marshall.
Goodness. I mean, I don't see why not. If you can get center of force, the beauty of the strideway itself is that you're able to physically get the stem cells and those pressures, and because they're so micro, they're allowing us to get multi-directional X, Y, and Z simultaneously, unlike other systems. I think you could probably do it with some of the insoles, but not my thing.
Yeah, we just had somebody grab the mic.
Yeah, I was just so the Tekscan walkway is just the hardware that the strideway replaced. So the answer I think is yes. It's just the classic walkway.
Excellent.
Check
Thanks,
with
Josh.
your local Tekscan rep.
No problem.
Thanks, Josh. All right, awesome. Marshall wanted to grab the mic too, so he's probably got the same comment.
Nice.
So in about a couple of minutes, I'm gonna have to go over and do some Jeopardy. But there's a ton of questions here. I'm gonna leave this up. Obviously, I won't stop streaming or anything like that. What do you got, Marshall?
Yeah, no, I was just jumping on to help Nick with that last question. It was, the strideway was our predecessor to, the walkway was a predecessor to our strideway. So the sensors are the same, more or less. The center of force data is calculated the same way. So you should be able to do the same. Yeah.
Awesome.
Great presentation, by the way.
Yeah. Are those turning patterns consistent across concussed persons?
Is the turning pattern similar?
Yes. With
Yeah,
concussed.
so some of my earliest work back in 2013, and I'm like, oh yeah, we're getting really back in the day now, right? We looked at the turning synergy in Parkinson's disease and in concussed individuals when doing like a 90-degree turn. But in general, if you're doing any type of turn, you always seem to go farther out than you need to and take more time and more steps. And your steps are actually wider apart and a bit more exaggerated because you have to get a wide base of support so you don't fall. And again, in some of our data, we think it's strongly linked to vestibular and or ocular based symptomology to start, which makes sense. You're dizzy, you can't turn very well.
Absolutely. Can we measure the effective length ratio by center of pressure with force plate? I think that's what that says, or from center of pressure by force plate?
If it can be derived from center of force, I don't see why not. We are limited on spatial temporal characteristics, so I think you can if you're doing a single pass. I don't think you can do it if you're going to do double passes because then you need to analyze each footfall independently. Gotcha. It does, again, you're talking post-processing, but the way we've set it up from our data, and I can only talk to the tandem gait data really specifically, we are able to run an automated MATLAB processing behind the scenes and get this data out in about 30 seconds for patients.